start.
Good morning, everyone, and thank you for attending this webinar. My name is JT Yeager, and I am Enartis USA's viticulturist and soil scientist. Today, we're going to be talking about the vineyard fertilizing product line, BluVite and BluVite Red, basically covering the concept behind it, and then look at some of the results from the 2019 growing season. But first, if you've never been to one of our webinars, I just want to go through some things before we get started, and just to let you know how they work. Firstly, we'll start off with a 45-minute presentation, and then we'll follow that up with a 15-minute question and answer at the end. This is a recorded session, so if you like the content and want to share it, it will be available at the end to listen to at a later date. You can also access the PowerPoint and PDF form and other resources by clicking on the folder in the bottom right-hand corner of the screen. Also, if you have any questions, please do save them until the very end at the question and answer portion. So make sure to have something handy to write down your questions so you don't forget them. Lastly, if you're having any technical difficulties, please respond to chat box 2, and Whitney will be there to help you. So here's the agenda for today, or this morning. I'll start off with an introduction to the product, basically looking at the concept itself. You really can't talk about BluVite without talking about soil biodiversity, and so I'll briefly explain the soil microbiome and how that works with the product itself. Then we'll move into the main portion of the presentation, and that is the trial overview itself. Basically going through specific examples, looking at these various parameters and including canopy measurements, juice analyses. Unfortunately, we don't have the preliminary wine analysis at this time, but we will have this available for distribution shortly. So now moving on to the introduction of the two products, looking at sort of the concept summary itself. So really, biological diversity is one of the main components in soils, and it's really essential for the proper functionality of the soil in terms of nutrient cycling. And any human-induced alterations to the soil, so tillage or any sort of management practice that's common in a vineyard especially, it can lead to an imbalance in the availability of nutrients, and we do this by disrupting these nutrient cycles with our management practices. If you think about it, the oak trees and the forest surrounding the vineyards, they don't need to be fertilized. So why do we have to fertilize our grapevines? One of the main reasons for this is we're moving from the system every single year with harvest, so we're taking grapes out of the vineyard. But there's also things that we do in the vineyard that are very disruptive to these natural cycles that I'm talking about. So really, the purpose of BluVite is to increase the soil's capacity or ability to function within this managed ecosystem that we've created in the vineyard. And really what it's doing is working with the native soil microbes. And so the general concept of BluVite really revolves around this idea of sustainable soil management. And it's becoming more and more, or increasingly more important with a changing climate. So just to elaborate a bit more on sustainable soil management. So again, BluVite is working directly with these native organisms that already exist in our vineyard soils. I just want to make it very clear though too as well, in parentheses here, we're not introducing any foreign microbes to the soil with the intent of changing this natural soil ecosystem. We're not inoculating anything. We are essentially just stimulating this already pre-existing plant organism interaction, and in this way we can actually improve the rooting of the vine and improve the vine nutrition, sort of in a natural way. And this is with the ultimate goal of reduction in synthetic chemical inputs, or synthetic fertilizer inputs, rather. We basically want to grow the best grapes we can, the lowest environmental impact possible. And again, like I said, this is becoming increasingly more important with the changing climate and more extreme weather events, and sort of as the future of grape growing becomes more uncertain. And the last point here I want to make, the ingredients are naturally sourced and non-GMO, and the product can be used as a part of many organic and sustainability oriented programs as well. So what is soil biodiversity and the soil microbiome? So I'm going to explain this a bit. So essentially, the soil microbiome is the living ecosystem that exists in every soil across the entire planet. And really, researchers are only beginning to understand really sort of the tip of the iceberg in terms of understanding its complexities. And you can see here, I mean, the numbers are pretty staggering. In any given vineyard, in only a gram of soil, you have over 10 billion individual microorganisms. And so that means in an acre, in an acre of vineyard soil, you're likely to find up to 2 tons of microorganisms existing in there. So for this last bullet point here, the most important microbial type is these root-associated microbes, both bacterial and fungal populations that work closely with the roots of the grapevine. So now moving into sort of the interaction that BluVite is acting upon. Really the takeaway from this slide is that there are some pretty complex interactions that take place in the soil between the grapevine's roots or plant's roots and these what we call root-associated microorganisms. Essentially, the soil microbial loop is a reinforcing cycle, really, that results in the emission of these secondary roots, so these fine lateral roots, and these roots are especially responsible for the majority of nutrient uptake and water uptake. And so these secondary roots play a very important part in the longevity of the vine and the vineyard, especially through extreme weather events like drought, because the vine can better utilize the water and nutrient resource. Microorganisms, obviously, in this bottom section of the loop, are a critical piece because they essentially take nutrients that are plant available, or plant unavailable rather, and the exudates that these vine roots release, basically high carbon rich compounds, and they take these unavailable nutrients, and through population turnover and predation from other microorganisms, these plant unavailable nutrients become plant available. So how do we know that BluVite is actually helping this plant microbe interaction? Well, we've seen the results. We've seen this change in the root structure and the root biomass. We actually did nursery trials, and what we did see was that the secondary root production that I'm talking about here actually was greater in the treated vines across the board, really. You can see that in the picture to the right, you can see the increase in that fine lateral root system. And maybe your next question might be, what are we doing to try to understand how the product is influencing this microbial community? And I have some answers for that as well. But sort of leading into that question, I first want to look at sort of the research setting, maybe just briefly cover some research that is being done. I think the study is being done at UC Davis. I'm not going to spend too much time here. The reference is at the bottom of the page if you want to learn more about this. But really, we're only just beginning to understand soil microbial biodiversity on a deeper level. And this particular study looks at the native soil microbial biogeography, if you will, in relation to Napa Valley AVAs. And the figure on the left, figure 1, just shows the American AVAs, American viticultural areas, rather, in Napa Valley. And then figure 2 corresponds with figure 1, showing the dominant taxonomical groups and their relative abundances in each AVA, given as a percentage, as you can see on the left. And sort of what they found was that each AVA has its own unique bacterial fingerprint, or soil bacterial composition, which show that biodiversity, well, basically which shows what biodiversity we can expect to find in a baseline vineyard soil, and really has implications for something being more and more discussed as soil microbial terroir, a unique microbial terroir in each vineyard soil. So basically, the research setting is really important for us to understand soils, but not really practical in the commercial vineyard setting when you're running field trials due to cost, but among other factors, speed in which you can get your results back. We have partnered with a company called BIOMEMAKERS, and they essentially can look at your soil biodiversity for about $200 a sample, and they give you a readout. This is the phone app platform, but they also have a desktop platform. But basically, they give you an idea of your baseline microbial population and how it might change over time as you implement new or more sustainable farming practices. And so we were actually working with a few larger growers here in California, and we're trying to better understand how exactly BluVite is influencing this soil microbial population. This is actually only our second year of collecting data with WineSeq, which is the name of this this platform. So we need to actually collect more data, probably a few more years of data before we can make any conclusions here as to how BluVite is affecting certain microbial populations. So now, before we get into the vineyard trials themselves, I want to introduce some specifics about the product itself. So we're really talking about two different vineyard fertilizing products, BluVite and BluVite Red. BluVite being for white grape varietals, and BluVite Red being for red grape varietals. On the product label, you can see here, I'm just going to go through some of the main points in terms of use and how it works. The product label does say it contains minerals, including elemental sulfur, magnesium sulfate, and yeast hydrolysates or yeast protein hydrolysates. And it does come in a powdered formulation, but it isn't entirely water soluble. So you have to actually maintain agitation as you apply it, so that some components of the product will stay in suspension. You do spray it in the vine row, and so this is really to maximize the coverage, the soil coverage, and ensure that it's uniformly applied. And then actually you see here that BluVite is applied in two early season applications, one at bud break or bud burst, and one at the beginning of flowering. And really this is so we can influence the vines early on in their growth and maximize the effects that we see later on in the season. This is just the application rate here, 16.5 pounds per acre or 18 kilograms per hectare if you're listening in from other places, maybe Europe. So that means really 33 pounds per acre is what we're looking at for the entire season. And depending on your growing region, you may get some rain events around bud break and flowering. And so application volume might be lower for you, 22 to 28 gallons per acre for wetter soils. And then for most situations, in California at least, you'll have to use between 65 to 86 gallons per acre, especially when you're applying to drier soil. I've seen pieces of equipment that can apply up to 100 gallons per acre. Really, if you're applying to dry soils, you want to maximize the amount of water that you use to apply it. So now moving on to the trials themselves. This year, we partnered with over 35 different wineries and growers in 4 different states. Really a good partnership with these wineries and Enartis USA. Data was collected from over 65 different vineyards with most commercially important varietals, were included part of this trial. A lot of Bordeaux varietals, a lot of Rhone varietals, Pinot Noir and Chardonnay. But do note that around 60% of the trials were done on Cabernet Sauvignon and Pinot Noir, essentially a 50-50 split between those two varietals of that 60%. Excuse me. So what measurements do we take? I'm not going to get into sampling logistics. If you want to see our sampling protocols, it's no secret. We can share that data with you. And essentially, if you want to mimic these trials that we did in your own vineyard, we have documents that you can follow. So what did we measure? Again, we did BUDBREAK measurements. This is for second-year applications only. We did shoot and canopy measurements, and both of these types of measurements were taken using a digital caliper and a ruler. The SPAD measurement is essentially a measurement that looks at chlorophyll concentrations and uses a specialized piece of equipment called a SPAD meter to do this. The juice analyses was mostly performed at Vincre Labs in Windsor, California. And unfortunately, again, like I said, we don't have the wine analyses ready for today, but we should have this available in December, hopefully. Also, just this last point here, we did have visual observations to support the data that we did collect. This includes fruit set observations, internode spacing observations, and we have some data on water stress symptoms that were very interesting from the trials this year. So now really kind of getting into the data itself. This is a Cabernet Sauvignon vineyard in Lake County, the Red Hills AVA. This is a spur pruned double cordon vineyard, and BluVite was applied in 2018 and 2019 here. So like I said, you really would only see the benefits in BUDBREAK after the first year of application leading into the second year. And at the beginning of the 2019 growing season, which is the second year of application, and looking at just after BUDBREAK, the length of the shoots were observably longer in the treated vines compared to the control, as you can see here. What was really interesting was the sort of distribution of shoot lengths. It gave us indication of increased uniformity in the treated area. So there's actually a decrease in the variation of the shoot length across the vineyard. So the shoots in the BluVite treated were more likely to be all the same size, whereas in the control you have longer and shorter shoots, and that variation is higher. If you remember from your VIT 101 classes back in the day, much of the energy source for shoot growth at BUDBREAK comes from the nutrition the vine was able to sort of store in its woody parts the previous season. So this does have implications for vineyards treated with BluVite, that they're able to actually better able to store nutrition in their woody parts the previous year. And here I have the statistic. I didn't include numbers for this. We have that data, but almost 11% increase in the shoot length overall at BUDBREAK. So this is actually a vineyard adjacent to the other property that I just mentioned. This was also, BluVite was also applied to this vineyard for 2 years in a row. And the same concept of nutrient storage applies here. If you know anything about cane pruned Cabernet Sauvignon, which this is, cane pruned Cabernet, it is notorious for something called apical dominance, and it makes it prone to having these non-sprouting shoot positions mid-cane, which you can see by these red arrows here, indicated by these red arrows. Based on the data from about 50 vines per treatment, we counted an average of 1.1 non-sprouting buds or non-sprouting shoot positions per cane in the treated, and about 3.4 in the control on average. So you're pretty significant reduction in blind buds per cane. And really this has implications for yield. I mean, you have to consider that there's in the untreated vines, the control vines, there are 2 less shoot positions in which clusters could form from. So now moving on to, well, actually this is to hammer home the point that I was making in the previous slide. This is that same vineyard. These might even be like the same vines, but you can see here that these non-sprouting shoot positions later on in the season leave these windows in the canopy. Essentially, this apical dominance becomes apparent. In the control picture, you can see that the longest shoots are at the basically the head of the vine, or basically those first shoot positions, and the very end shoot position at the end of the cane. In the BluVite Red treated on the right hand picture, you can see that there's more uniformity of the canopy. You're not seeing this window that I'm speaking about. So enough pictures here. Let's see some data. These 2 charts are comprised of thousands of data points that were collected by Enartis USA, and some data points were collected by the growers themselves too as well. A lot of vineyard management companies, a lot of bigger wineries that have vit teams that helped out with collecting this data. These two charts are comprised of all varietals, all AVAs, all counties in California and Oregon, so it gives you a pretty wide data set, basically across 3 months of data collection. You have shoot length in centimeters on the left, and then you have shoot diameter in millimeters on the right. And so really the point of this slide is to show that the distribution of shoot length and shoot diameter gives you the sort of increased uniformity and strength of those primary shoots. You can kind of see this. I kind of wish I made a little distribution curve here, but you can see this upward shift in the distribution of shoot length and also in shoot diameter in the BluVite Red treated vines versus the control. And really, you can tell by these peaks that the shoot length and shoot diameter were actually more uniform across vines with fewer shorter and thinner shoots in the BluVite Red treated. And I left some numbers down here. Median length for BluVite in this chart is about almost 100 centimeters, which is 15 more centimeters above the median average for the control. And median diameter, we're looking at about 1.5 millimeters greater in diameter between the two. So same thing here. I don't know if you remember from my introduction slide, but the SPAD measurement or SPAD indices is a measurement of photosynthetic compounds or chlorophyll. And so we took a similar amount of measurements, not quite as much as the canopy measurements, but it's a couple thousand data points here. And you can see this, again, this upward shift in the distribution of SPAD measurements in the BluVite Red treated vines. This does give an indication of a great amount of chlorophyll and photosynthetic activity. And this is important, especially early on in the season, because the vine will have nutrition to devote to the clusters early on in their development, and it does also have implications later on in the season as well for cluster maturity and ripening. So just bringing it back to some more visual observations that we made here. We did take some measurements as well in terms of leaf diameter. We did measure internode spacing as well, but I'm not going to cover that. I can always share that data if you're very interested in that aspect of it. But this is a Zinfandel head train block in Sonoma County. And in the left-hand picture, you can see that there's leaves that were selected for their higher SPAD measurements, which again is that photosynthetic measurement, the measurement of chlorophyll. And so you can see that there is actually observable difference in the shade of green on the leaf blades, and there's actually a darker shade of green seen in the BluVite Red treated vines, which does suggest, among a few things, an increase in leaf maturity and an increase in chlorophyll content, these photosynthetic compounds. And again, the SPAD data does support this on the previous slide. In the picture on the right, we see these are the shoot apexes, and we can tell from this image that well, basically there's more active shoots. You see this in sort of the growing tips themselves, the shoot apexes, and sort of more active tendrils. And what I actually found interesting about this was that that there was more homogeneous internode spacing relative to the control, and this has pretty huge implications for the activity of the root system. If internode spacing is uniform across the vineyard, then really the activity of the roots is uniform across the vineyard. If you see variation, then there's variation in root activity. Oh, just to mention, forgot to mention about this. This is some of the measurements. We would take leaf diameter, but it's hard to kind of show those measurements here. So I just drew a circle here, and you can see that overall leaf diameter on the two largest leaves from each treatment is greater in the BluVite Red. We also took petiole measurements too as well, and leaf thickness measurements, and found all to be following that similar trend of increased size in the BluVite Red treated. So this is kind of to show you what I mean by a shoot apex here. So this is a Pinot Noir vineyard in Polk County in Oregon, and this is a cane pruned Pinot. And this is really just to hammer home the point that this second hang wire is marked in red. And although there are shoots that make it to that second hang wire in the BluVite, or actually in the control rather, they're actually far fewer and much rarer when compared to the treated vines. You also begin to get a feel for increasing uniformity here, where you can see the bottom picture. There are fewer shorter shoots, and there's an increased number of shoots that reach that second hang wire in the BluVite Red treated. So really, we're not increasing vigor past what the vine can do. Really, what we're trying to do is those shorter shoots that don't quite make it to that second hang wire where you want to be. We're actually allowing that vine to fill out the canopy, and this does have implications again for how much resources the vine has to be able to develop to cluster ripening. So briefly look at fruit set here, and this is first year application, BluVite and Pinot Noir. This one is, so we're now back in California, and this is a cane pruned Pinot Noir vineyard again, Russian River Valley AVA. And you can see, we have seen many examples of this. I'm just including one here, but we consistently saw better and more even fruit set per vine. And we know this because we did pretty extensive berry counts, essentially plucking berries off of each cluster. Hundreds of clusters, maybe even close to 1,000 clusters we plucked berries from throughout the whole season, myself and the lab that we worked with, Big Free Labs. And we consistently saw the better fruit set with less variation in the clusters in terms of the amount of berries that we found. Here you can see this specific example shows that we had on average 23 more berries per cluster in the BluVite Red treated versus the control. And you can kind of see that from the picture as well. So now looking at pre-harvest here, this is Cabernet Sauvignon vineyard. It is spur pruned, and it's in the Stag's Leap District AVA in Napa County. And just to sort of reinforce the point I was making in the last slide, we measured that there was on average 56 more berries per cluster. This is sort of a more extreme example. In this vineyard in particular, there was a rain event in May that occurred, and it really had some pretty huge implications for fruit set. And so I think this is why you're seeing such a high increase in the number of berries per cluster here, 56 more. And this really equated to 58 grams on average increase in cluster weights. And you can see here, there aren't any significant increases or differences in average berry size itself. So here's just one example of sort of the differences in water stress that I was speaking on. This is a Cabernet Sauvignon vineyard. It is a cane pruned, and this is in the Oakville AVA. And what we did here is we looked at a total of 200 vines for incidence of water stress, and we actually tallied those for the presence and absence of basal leaf chlorosis, which is the yellowing of the basal leaves, which is typical of water stress. We did work with some growers that took pressure bomb and stem water, leaf water potential measurements to back sort of this up as well. But I thought that the yellowing would be a good visual. And what we did find here was that over half the vines showed signs of water stress in the control block, and only about 10% of the vines showed it in the treated. So there were vines that showed water stress in the treated, but there were far fewer of them. And also, sort of to add to this, what is interesting about this is this came along with berry shrivel as well. What is interesting about this is this came along with berry shrivel as well. So we saw a higher incidence of berry shrivel in the control block. In that 50% of treated vines, you're more likely to see this sort of image on the left here. And this has pretty huge implications for reductions in yield, but also for fruit quality. So now to move on to the juice analyses, and the more pale gray colored in both graphs, the more pale gray and pale red colored is this Pinot Noir vineyard in the Russian River Valley, and the darker gray, darker red color is the Cabernet Sauvignon vineyard in Adelaida district in Paso Robles. And the Pinot Noir vineyard had only had 1 year of application. The Cabernet Sauvignon vineyard in Paso had 2 years application, both 2018 and 2019. And basically what you can see here is yeah, on the final sample date, the potassium concentrations in milligrams per liter was actually only slightly lower in the Pinot Noir vineyard. But the second year application, you can see that really where the differences start to pull away. I think it's almost, if I can count here, almost 400 milligrams per liter less potassium in the juice. And so this is particularly important really to lower potassium in grape juice because of improvements in the buffering capacity of the resulting wines and actually the cold stabilization in the finished wines themselves. On the graph on the right-hand side, you can see that there aren't huge differences in pH. And really what I think this comes down to is that after fermentation, the pH of the resulting wines will principally be determined by these grape juice potassium concentrations. So once we get the wine data back, I anticipate this is really where we're going to see the differences in pH. So this is actually kind of a cool trial that we did. This is actually 1 and 2 year side-by-side in a Cabernet Sauvignon vineyard that we were able to do. They saved a section of the block for the control, and then they had a 1 year application that was done in 2019, and then they had a section of the block that had it applied in 2018, and then they made the application in 2019. So control 1, 2, side-by-side. And it's following a pretty predictable trend here. We really did see that clusters had higher average weights in each year of treatment. And this is in this particular example, clusters had slightly larger size berries in the 1 year and the 2 year treatment. But what we found was that there was a significant increase in the number of berries per cluster. The average here isn't drastic, but really what you can see here from a vineyard that does not have issues with fruit set, we were still even able to increase in the average number of berries by well, nearly 10. That's like 7 berries per cluster, which isn't huge, but if you consider that across an entire vineyard, of 150 acres, that has pretty serious implications for yield. So here, looking at sort of anthocyanin content, and I want to point out that in this first chart, that's the same vineyard that I was speaking about in the previous slide. And what you can see here really is that the chart on the left, in just the first year of application, you're not seeing huge increases in anthocyanin content, but we have already increased the amount of berries per cluster and cluster weights. So you're increasing yield, but maintaining quality. In the second year application, not only are you increasing the number of berries, increasing fruit set, but you're actually significantly increasing the total extractable anthocyanins within those berries, within those clusters. It's pretty exciting. So the graph on the right, this is again the comparison of the Pinot Noir vineyard in Russian River Valley with the paler gray and red, and then the Cabernet Sauvignon vineyard in Paso Robles area in the darker gray and the red. And so really you can see that this follows the same pattern really. We see slight increases in the first year application, about 150, maybe 200 parts per million increase in the Pinot. And then the second year application, really you see the two pull away. I think this is one of the more extreme cases here, but we saw an increase of 700 parts per million of total extractable anthocyanins. So I was kind of having a hard time figuring out where to put this slide, but I think at the very end is nice. Fast forward about a year ago, and you see that this is a Cabernet Sauvignon vineyard, actually the one from the second slide of the field trials. The one that has issues with apical dominance. And you can see here that on average pruning weights in this chart on the right, or the left rather, that pruning weights were 13% higher on average in the BluVite Red treated vines when compared to the control. Also, you see from this first picture that shoots found at the beginning and the end of the canes were much longer in the control. And you can see actually the length of the control is larger, and that's because of these shoots at the beginning and the end of the cane. And you see sort of this apical dominance that I'm talking about. In the BluVite Red treated vines, you can see that the shoots aren't as long, but you actually see that more of them are sort of the same length. So you have implications for increases in uniformity, which means that there's less difference between the lengths of shoots at any given position. And I don't know if you can tell from this picture, but it was really apparent from when I was taking the pruning weights and pruning the vines that there was actually a significant decrease in the color of the pruned wood. We actually saw darker color of the pruned wood in the BluVite Red treated vines, which does have indication, does indicate better lignification in the shoots during the season. So we're going to do some follow-up field trials experiments with this to see if we can quantify this a little better. So here is a summary of the benefits of using BluVite. Really, the product favors primary shoot growth and more balanced vine growth. We get an increased bud sprouting on mid-cane positions in cane pruned vines. As I've mentioned, we get increased SPAD indices and chlorophyll content, photosynthetic activity. We see more even fruit set per vine and less variation in fruit set between vines. We also get an increase in the average number of berries per cluster, which was apparent from the data. We also saw an increase in average cluster weights themselves, and this is even with first year of application. We did see decreased concentrations of potassium K, or potassium levels rather, in the grape juice, and really this effect was seen mostly in the second year of application. And then we did see increase in the total extractability or total extractable anthocyanin content in the grape juice itself. So thanks again for attending the webinar on BluVite, and remember to please answer that survey at the end when you leave. Any feedback will only help us to make our content better for future webinars, so please fill out that survey again as you leave the webinar. Please ask your questions in that box now as we begin our question and answer portion of the webinar. Thank you.